Hello fellow biogeeks. In this video I'm going to talk more about the rough endoplasmatic reticulum, the smooth endoplasmatic reticulum, and the Golgi apparatus. This just gives like a nice overview of what the cell looks like and what all the organelles are. In my previous video I talked about the nucleus and the membranes overall of the cell. This time I'm going to talk about three organelles at the same time because just the endoplasmatic reticulum together is the RER and the SER together and the Golgi apparatus, and they have like an interplay between each other. So I thought it would be good to explain these three at the same time. So first I'm going to talk about the rough endoplasmatic reticulum. Maybe you can already see why it's called rough, because it has ribosomes on it. And what are these ribosomes important for? Well, the rough endoplasmatic reticulum is like bound to the nuclear membrane. So this gives like easy access of the messenger RNA that's produced inside the nucleus to go to the rough endoplasmatic reticulum. It's very close by. So then these ribosomes will like bind onto the messenger RNA, like you can see here, and it will translate it. So this means that it will scan the messenger RNA, and as it scans it, it creates amino acids. And at the end, there's like a stop codon, so then the amino acid chain will be released. However, this amino acid chain or this protein is not folded correctly. So what the RER does in addition is it also folds these proteins and will also perform post-translational modifications on it, like for example phosphorylation. And when there are misfolded proteins, that can always occur. The cell is not perfect. So when that happens, the RER also like degrades those proteins because these can be harmful to the body. And what the RER does in addition is it stores calcium, and this can be important for signaling pathways, for example. So this is what the RER is important for. Next up, the smooth endoplasmatic reticulum. It's important for other things, like not really in protein synthesis or something, but in, for example, the lipid synthesis. And what does it synthesize? Well, it can synthesize phospholipids and cholesterol. It can do more, but these are just two examples. The phospholipids, as if you watched my previous video, the phospholipids make up the membrane, the plasma membrane, and it's a bilayer. So it's very important to keep these phospholipids and keep synthesizing it because a lot of these phospholipids are needed. But they also make cholesterol. And you might think that cholesterol is a bad thing because maybe in advertisements they say that you should keep it as low as possible. However, cholesterol is still very important because it sits in between phospholipids, and this creates a more fluidic membrane. So this means that, for example, with migration, the membrane can like reshape itself a bit so that it can migrate. So cholesterol is important, but again, if it's like too low, you should be careful with that too. Then the cells get like more rigid. They can't really reform itself that well. So cholesterol is important, but of course too much or too low from it is not good. Then what you can see here is the adrenal cortex. And the adrenal cortex has cells that have like smooth endoplasmatic reticulums that are important for synthesizing steroids. This is the same for gonads, so testicles or ovaries. In these gonads, they also have cells that are important for the production of steroids or steroid hormones. Then lastly, if you have like toxic drugs inside of your body, the smooth endoplasmatic reticulum is important for degrading those by, for example, making them more water soluble so they can be detoxified more easily. And it's same as the rough endoplasmatic reticulum, the SER also stores calcium within it. And then we go to the Golgi apparatus. And you can see that the Golgi apparatus has two sides. It has like a cis side, so this is a cis side on top, and at the bottom it has the trans side. And you can imagine that the rough endoplasmatic reticulum is situated over here at the top, and it will send out its proteins in vesicles. So so the rough endoplasmatic reticulum sends out proteins, and these are like surrounded by a membrane. And this will be transported towards the cis side of the Golgi apparatus. Be careful with that, it's always the cis side, because at the cis side it will fuse with the membrane of the cisternae of the Golgi apparatus, and then the proteins inside it will be released inside of the cisternae. And in the cisternae, there will be some modifications like sulfation, glycylation, and phosphorylation, and a couple more. However, these proteins will then be transported towards the trans site of the Golgi apparatus, and at the end, the proteins will be released again inside of a vesicle, so again in a membrane. And these vesicles will then be transported to its desired destination so that these proteins can exert its functions. I find this very interesting that it has like it's almost like the industry. If a product is made, you have first the nucleus that will produce the nucleus that has genes. These genes will be transcribed into messenger RNA. Then this messenger RNA will go to the rough endoplasmic reticulum, and the ribosomes on this RER will translate this messenger RNA into a protein. This protein will then be packaged like in like a factory. It will be packaged, will be sent towards the Golgi apparatus. It will only be sent to a certain side of the Golgi apparatus, the cis side. Then it fuses its membranes with it, and then it will undergo modifications, and it will finally leave the trans site. And then the cell just knows where this vesicle has to go for its function. I just find this very interesting. But yeah, these are like the three organelles I wanted to talk about. I hope you found this very interesting, and if you did, I will always appreciate a like on the video. And if you happen to misunderstand something out of this video or just a random other biological process, you can always go to the link in my description and order a private tutoring session with me so that I can prepare your question and explain it to you in much detail and as well as possible. So thank you all for listening, and I'll see you in the next video. Bye, BioGeeks.